right, so good morning, good afternoon, welcome in across the country and beyond, everybody. My name is Jesse. I'm with you here at Exploring by the Seat of Your Pants. And for those of you who are joining us for the first time, like our speaker today, we are all about bringing conservation, adventure, and science into classrooms around the world. And I am so pleased of all days in the year that you are joining us today, because today, June 8th, is World Oceans Day. We have been celebrating World Oceans Day as an epic week-long celebration since we were founded back in 2015 with a huge blitz of incredible events, and this year is no different. We have done 21 broadcasts by the time Friday wraps up with amazing scientists and explorers around the world, and I'm particularly pleased too because this wraps up our epic mini-series with the National Research Council of Canada. On behalf of Ocean Week Canada, we've been partnering with the Canadian Ocean Literacy Coalition featuring their 180 plus events around the country. We had the chance to partner with the amazing NRC Canada team to feature some of their coolest scientists working on amazing projects coast to coast to coast. So a big thank you to the NRC. Check out the entire mini series here. Over 1,400 kids registered combined for these 4 programs. So it's been a really special and amazing journey. Now today we are going to be joined live by Jorg. Him and I did some amazing tech troubleshooting before this broadcast. So it's been a long time since I've had so much fun getting things ready before we went underway. And today he's going to talk to us today about going out on the ocean, what it's like being on a scientific research vessel to do incredible work. Specifically, their team went to look for phytoplankton. So the underpinning, the basis of the world's ecosystems are with phytoplankton. 1 in every 2 breaths you take come from oxygen generated by phytoplankton, and they serve as the foundation of so many incredible food webs and chains. So without further ado, we're going to learn what it's like to be out in the ocean. Jorg, thank you so much for being here today, man, and welcome to the broadcast, and take us away.
Yeah, I would do that. Yeah, now Jesse already took like quite a few things away that are actually so exciting about this kind of stuff. But yeah, I want to talk to you or with you about like how it is to be out on the ocean and to actually work on a research vessel, on a research boat. And so this photo here, for example, I took that photo in the Bay of St. Lawrence, and it's like really, really early in the morning here. It's like 3:30 a.m. It's just like the very first light comes up, and we are already up and working, because usually when you're on a research boat, you are out on the ocean and it's like time is money basically. So you want to work. And phytoplankton never really sleeps. So you want to also maybe find out what happens in the day, what happens at night. And so you're basically working the whole time. This weird thing that's coming out of the water here is something what we call a CTD. And this instrument will be a little bit the focus of my talk, because I want to show you guys what we do with it and how it's basically incorporated into the research work that we do. But first, just a little, let's see if I can get from one slide to the next.
A lot of work. After all our work to get here, it's going to, I believe in it. I really do.
I know. What is going on?
Oh, no.
Oh, here we go.
Perfect.
Okay, maybe just a little delay. So first, a few words about me. So that's me actually on the boat and up. I was up here taking the photo. This is the CTD I was talking about. And I'm taking water out of the CTD and putting it into like a lot of bottles. But I will explain all these things in a little bit more detail throughout the next like 15 to 20 minutes. So my name is Jorg Bienke, and I'm we call it here at NRC, a research officer. And I work in the aquatic and crop resource development. And I'm not from Canada, as you might maybe hear in my accent. So I'm actually from a city in northern Germany, and the city is called Kiel. It's right here, and we are also right next to the Baltic Ocean. Our province is actually also surrounded by the North Sea here on this side, and we have like a lot of really beautiful beaches in our area close to the city. So I spend a lot of time on the ocean, and that's, I think, also why I fell in love with the ocean, and I cannot imagine not living next to the ocean. This would be for me like impossible to think, even thinking about it. And my hometown is really, really well known for a crazy sailing festival that's happening every year. And you can see here, it's like the tall ship parade, and you have like hundreds of boats that are just like in this bay here. And my hometown is basically here in the back. So first, I mean, why are we so interested in the ocean? And a lot of you or some of you might already know some of these facts. But first fact for sure, 70% of the Earth's surface is covered by water. So that is really a lot. So, we on the land think that it's so important to be on the land here, but actually there is way more water on the surface than land. And 40% of the CO2 that we release into the atmosphere, like we call it the anthropogenic CO2, from driving around in the car, flying all over the place, like making all kinds of stuff, like 40% of all of that CO2 has actually been taken up by the ocean. So it's like a super important part to keep our Earth basically healthy. And the ocean, therefore, also controls the Earth's climate. And this is kind of like the first figure that I wanted to show you guys. And this is called the ocean conveyor belt. And this is one of the most fascinating things that is actually happening in the ocean all the time. And some of you might know the Gulf Stream. It's like a very warm water stream that goes up here into the North Atlantic. And while it goes up, it starts to cool down. And what happens when the water cools down? It gets denser and it gets heavier and it starts to sink down. This is actually not really correct. It more sinks down up here. But anyways, the water starts to sink down and then it goes on the bottom of the ocean and goes all the way around here. Here you have Antarctica, so it goes through the Southern Ocean and it comes up in the North Pacific. And then from there, the water goes all the way around again until it's back into the Gulf Stream to then go back down again. The whole conveyor belt takes around 1,000 years. So that's why we really have to be careful. Whatever we do now might have also consequences like later on and way later on. So the ocean is not only connected with each other, it's also, there's always like a time delay. If you do something here, it might take a long time, but at some point you might see the effect actually here. Another crazy fact about the ocean is that the ocean is in average 3.5 kilometers deep. So it's a ginormous space for life because life happens in the ocean everywhere. And so you basically have to imagine you would walk into a forest and all the trees are 3.5 kilometers high because that's basically the biosphere that we live in, right? It's just like maybe as high as the trees are. And so if you want to compare it to the ocean, the trees would have to be 3.5 kilometers high. So this is really amazing. And I mean, beach life is just too fun, right? So we really should watch out what's going on in the ocean and we should find out how the ocean reacts to certain things. And that's also basically what we are doing here at the ACRD. So we want to find out how the phytoplankton basically react to certain things. And we went on 3 research cruises in the last few years. And so when you have here Canada, we are actually just here in the Maritimes. And that's also where our research cruises were. And research cruises often have like funny names like T-Rex 2 and T-Rex 3 and Cougar. And so this was actually the 3 research cruises that we were on. And so this cruise was with a boat that's called the Coriolis 2. It was here in the Bay of St. Lawrence. That's also where I took this photo. And then this cruise here, we went on like a pretty old, it's already now retired and being disassembled actually. It's called the Hudson, like an old Coast Guard ship. And then on this cruise, we were on a offshore supply ship where we put containers. You can see here these containers. And in these containers were basically our labs. Like on these boats, the labs were on the boat. And here we brought our lab onto the boat. Unfortunately, on this cruise, the boat, the propeller kind of like broke when we were around here because we wanted to also go up here, but we had to turn around and then go back to Halifax without actually really doing much science. So what is our mission? What do we want to do actually on the ocean? So understanding the response of phytoplankton to environmental stresses for the development of phytoplankton-based biosensors. That is so easy, right? So wait, no. What is this guy talking about? What is he talking about? So let's slow down and we do like step by step. So I'm talking about phytoplankton. So what is phytoplankton? And in this little abstract that I put together for this talk, I was talking about unicellular organisms. And so it's basically phytoplankton is mostly just like one cell, like this cell here. I actually took this photo with a microscope. And you can see here, we call this a scale bar. So that shows you the size of this, this phytoplankton cell here. And so this is 10 micrometers long. So what is 10 micrometers? I don't know if some of you also watched the plastic talk. 10 micrometers is really, really little. So if you have a meter, there are 1,000 millimeters in a meter, and then there are 1,000 micrometers in a millimeter. So this is really, really tiny, and you definitely need a microscope to see that. But the fascinating thing is, once you have a lot of them, and they come in all kinds of shapes and forms, and sometimes they stick together and they build chains. And if you have a lot of them, you can even see them from space with the satellite. And this here is also in the Baltic Ocean. Here you have Finland on the right. And so this color that you can see here in the ocean is actually phytoplankton, and we call it a phytoplankton bloom. So much to phytoplankton. But what are environmental stressors? That's such a weird word. But environmental stressors could be something like oil, for example. That was a project that I was working on to look at the effect of oil on phytoplankton. And this photo here is from the Deepwater Horizon catastrophe, which happened in the Gulf of Mexico, where millions of liters of oil entered into the ocean, and it was like a huge catastrophe. But environmental stressor can also be like garbage dumps, or it can be sewage that gets into the ocean. It can be like harmful chemicals that get into the ocean, and that just basically they are stressing the phytoplankton. So the phytoplankton, often then start to like decline, or they don't want to grow anymore. And that's basically what we want to figure out. And a biosensor. So a biosensor, because these phytoplankton, they react to these changes. So something comes into the water, and then the phytoplankton reacts to it. And we want to make a little chip that can measure this change, so we know when something is wrong. So it's basically kind of like a detector that detects the whole time what is going on. And once in a while, the detector goes completely crazy, and then we know, oh, something is going on and something is wrong. And so how do we do all that? And it all starts because we want to build this sensor or this detector for the ocean. So we have to go out on the ocean and actually do what we call incubation experiments out on the ocean with the phytoplankton that are there. So incubation experiments, it's a little bit like pretending something is wrong. So this photo here is from the Hudson. That was the Cougar cruise. And down here are like two colleagues that did this experiment here. And so here you can, for example, see there are a lot of buckets that they put up here on deck. And these buckets are actually filled with ocean water. And then you can see most of these buckets are actually clean, but some of these buckets are also really dirty. And these dirty buckets are actually buckets where we put oil into the ocean water. And the clean buckets are the buckets where we did not put anything in it, because you always need something what we call a control, because whatever change is happening in the bucket with the oil, you want to know if this change did not happen here. So it's like an oil-specific change that is happening. So this is my colleague Caroline, and that was on the Coriolis. And there I did like an oil experiment similar to this one here. And she actually did a different experiment. And so you can see here all these flasks, and they are also all filled with ocean water. And then you have like all these tubes in here. And we basically bubbled CO2 enriched air into these bottles, because we wanted to know, or she wanted to know, what happens to the phytoplankton when there is a lot of CO2 in the water, because the ocean takes up all the CO2 that we put into the atmosphere. So it's important to understand where the phytoplankton, how they will do in the future when there's even more CO2 in the atmosphere and even more CO2 in the ocean. So how do we get the water then from all these different depths? And that's exactly where this instrument comes into play here. And it's called the CTD instrument, where the CTD stands for conductivity, temperature, and density. So temperature, we all know, that's just the temperature. So it measures the temperature when it goes down. Conductivity is kind of like the salinity. The higher the salinity, the higher is the conductivity. And then you have the density of the water. So basically how densely, how heavy is the water. And so this kind of instrument, we use this to basically get the water from the ocean, because we cannot just throw a bucket into the ocean and then get the bucket back on board. That would be not really safe and not really a really nice way to do it. So how does this CTD then actually work? And you can see here that's where we deploy the CTD. So we will send it out into the ocean, and you can see it's also pitch black here. It's dark. It's at night when we do that, and you can see here all these lids are actually open, and you can see here these are also all open. And if we go back to the other slide, this CTD actually just came out of the ocean, and you can see all these lids here are closed. And you can see it here also a little bit more detail. So these are the lids that are open, and every lid has like one of these steel strings attached to this middle. And so because all these bottles are open, the CTD goes down into the water. And whenever we want to capture some water, we say we fire a bottle. It's called firing a bottle. And basically the string here is being released, and flop, the water that is in this bottle is being trapped because the lid on top and on the bottom closes. And so when you throw the CTD in the water, you can see how all the air just like gets out of the water. Always is a nice view. And then the CTD is in the water, and then all the scientists are going to the screens, and they are looking at like 500 different screens, and everybody's getting excited. What is happening down there? Because the instrument actually measures what's happening in the ocean in real time. So you see live basically what's going on. And then you see things like that. And we scientists, we are always like super excited about this kind of stuff. We are really looking at this data, and every meter the CTD goes deeper. And you can see here, that's the depth basically. So we went here like almost to like we went to like 320 meters here. And then you can see different, we call it profiles. So in blue, for example, that's the temperature, and then the temperature scale is down here. So you can see on the surface we had like 10 degrees, and then when it was like around 50 meters deep, the water was almost only 0 degrees. And then the water warmed up again, and here it was around 7 degrees. And so you can see all these little different things. And we know that something is going on here, and we call it like there are different water masses that have a different characteristic. And you see all these red lines. These are the lines and the depth where we say fire a bottle, fire another bottle, because we are interested in what is going on in this water here. So once the CTD is back on deck, then something's happening. We are sampling the water. And on this cruise here, we were also together with a crew of chemists, and they were doing like all kinds of stuff. They had like a lot of different bottles and different vessels. And here somebody's filtering the water. Here it's a glass bottle. Here it's a big bottle. There was a lot of different stuff happening. We, the biologists, usually do our incubation experiments. So we just fill up our bottles with the water, and then we put CO2 into the water, or we put oil into the water and incubate the water, right? Like I showed already this picture here with all these bottles. So these bottles are all prepared here. And that's then how our experiment looked at the end from the outside. So here is actually my experiment, because that's where I put the oil in the water. And then this experiment was Carolyn's experiment, where you have like all these tubes that went into each bottle, and then we were bubbling the bottles. And you can see here we used a blue screen, because the water that we use in these bottles actually came from like 5 or 10 meter depth. And you can imagine that if you like a little bit further down in the ocean, there's not so much sun coming down. So we protect this water a little bit from the sun by putting a foil on there. And you can also see that these incubators, that's how we call them, they are attached to the deck. And that's important, because sometimes the boat also looks like this, and whatever is not attached, it just rolls down, falls into the ocean, and most likely you will never see it again. Maybe some fisher will find it at some point. Okay, then after the incubation experiment, there is a part where we filter our water, because we want to analyze what is in the water going on. And these 3 things here, these are actually pre-filters, but it basically still shows you how the filter most of the times will look like. It's kind of like a brown slash. Sometimes it's a little bit more green. And that's then where we take the filter back into the lab, and then we start to extract the DNA, and we look what is going on. And then we try to make sense of it all, basically. And this is how our workbench looked on the research boat. So it's quite tiny, and it's a little bit small. You have some freezers down here. You definitely want to have good music. That's really important if you work day and night. And then you have here, it's a pump that pumps the water from the incubation bottles here into a sink. Over here is a sink. And then while it pumps the water over, the water goes through a little filter, a little bit like this. And then you capture all the things that are in the water. And that's what's basically it. Like, it really is very exciting to work out on the ocean. And I always love it when the ocean gets a little wilder, because then you can really feel the power that Mother Nature basically has. And here is one of my favorite photos for sure, where you just see like a huge wave, we were looking right out of the lab here onto the ocean. And sometimes the wave also gets so crazy that they are splashing on board. And sometimes the ocean is so crazy that you go back into your cabin and you think you have the feeling that you are in a washing machine. But that's all part of the big fun. Any questions?
Oh, so many questions. Well, if you want to come out of screen share, see us again, have a bit of a conversation. That was fantastic, man. One of our first questions, just via email, actually, before we go to our live classes, we got our 3 groups live on camera, a bunch of YouTube groups as well, is it ever dangerous? I mean, you highlight these big waves, you highlight water coming onto the deck. Are you ever fearful about the safety of the ship, or are they pretty well, good out there on the water?
No, the boats are in very good shape. Like it sometimes it might look dangerous, but you feel very safe. And also there's always the crew and there's the captain, right? And they do that every day, all the time. And they are always very calm. And it's when it gets just a little wild, it's pretty fun to be on the boat. At some point, it can get a little scary when things start to fly around in the kitchen. Yeah. Or when you have to hold your plate the whole time, because otherwise when you eat and you don't hold your plate, the plate just flies away. But overall, you do feel very safe on these boats.
Yeah. I'll note that we've had a bunch of Antarctic explorers over the last few days in our broadcast, and the water to go from South America to Antarctica is some of the choppiest in the world, and none of them feel ever in any danger, even in huge swells, hurricane force winds. These boats are designed to be very, very resilient. And there's tons of safety protocols in place if something ever were to happen to make sure that people would all get off okay. So I'm glad we got that question. Thanks, guys. Second question online that I'm going to head to our St. Monica Grade 8 crew, Ms. Lana and Ms. Randall, stay tuned, I'm coming to you soon, is about how long the research cruises are. So you've talked about going on a few of these in the last few years. Is there a standard length? Is it a few days, a few weeks? What's the timeline like?
Yeah, it always is. It's depending on what you're actually looking at, and also it's depending on where you want to go to look at it. So the cruises that we were on here, they were usually like around 10 to 14 days because that was the time that it took us to basically do this trek. But I was like in 2011, I was involved in a cruise where we actually were on the water for 4 weeks. So that's already a little bit longer. We went there from Cape Verde to Gabon. That was like in the tropical Atlantic, which was really beautiful. And then there are other cruises, sometimes with German research vessels, where they go all the way from northern Germany all the way down to basically Patagonia, Argentina, and that's then like a 6 weeks cruise where you're on the boat. The crazy thing is really when you are like when we were on the Atlantic cruise for 4 weeks, we only saw one container ship in 4 weeks. Otherwise, we were just by ourselves the whole time.
Cool. How neat is that? Thank you for that. No one's ever provided that sort of context on what the water is actually like. And for kids that are keen too, our first speaker during Oceans Week is going on a 2-year cruise with her crew, which is a little bit long. That's very dedicated to want to be on the water quite that long. But great question, guys. St. Monica, Ms. Whaley's class, grade 8. If you want to unmute your mic, come on in. You should be good to go. We're hoping the audio is going to work. Fingers crossed. And then if worst case scenario, you will be able to share in the chat if it doesn't want to work. But if you want to have a question, come on up, unmute, and we'll head to Ms. Lada's class in a second next.
Can you hear
me?
Hey. Are you
Perfect. Yeah, we can hear you.
Yeah, okay, I'll relay your question.
Sure. I just wanted to know a bit about career-wise. So what you studied, where, for how long, how did you get into this field of work?
So yeah, career-wise, how did you get into this field of work?
Great.
Like, how did you study in school and so on?
Yeah, that's a good question. So I was actually I wanted to be a marine biologist since I'm a little kid. And then in grade 10 and grade 11, when we suddenly had like molecular biology in school, I was really fascinated by that. And so I did a master in biochemistry actually. And but I always wanted to go into the marine field. And so my hometown, we have the Geomar Institute, and they are like very well linked to the university. And so during my master, I already did like quite a few courses, like at the Geomar Institute, where I then did like marine chemistry and marine biology. And then I did my PhD actually here in Halifax in Canada in basically like cell biology and phytoplankton. And so I'm more a cell biologist, so I don't go on cruises all the time. There are like the chemical oceanographers. They are out on the ocean like way more often than us, because they have to measure basically all these parameters in the ocean. But if somebody really wants to go out on the ocean, like oceanography, biological oceanography, chemical oceanography, physical oceanography, these are like kind of like the things that you want to then go towards to.
Well, and this is from the science angle too. We just wrapped up with the Nautilus crew a couple hours ago, and they highlighted the fact that if you're on a research cruise or any sort of scientific enterprise, there are captains of the ship, there are communications professionals, roboticists, engineers. I mean, we talked about the chefs on the Nautilus. So if you're interested in being involved in a grand natural or scientific enterprise, whether it's oceans or Antarctica or NASA, there are thousands of roles available to you that do not necessarily require you to have the education that Jorg has, that some of the other people that we've had on. If you want to be a scientist, great. As you heard today, scientists get to work with really cool people in really cool places and generally have a big smile on their face half the time that they're doing the work. Which is
there are also actually CTD technicians, for example.
Yep.
Right, that are really just specialized on operating this CTD that goes up and down. And I think the Coast Guard is always looking for people, basically, like as you say, also chefs and deckhands and because I mean, but that's a tough job, right? That's really, then you are on the ocean all the time, and you really have to be born for it, I think.
Yeah, great answer, guys, and great question. Miss Lattice class, I'm going to head to you guys next. Miss Randall, I'm coming to you right after. If you want to unmute your mic, you're good to go for your virtual school. Hey.
Thank you. Yeah, so you can just see me, but behind the screen there are like 47 other students. So hopefully you can hear. All right, Anvi, you're up.
Wants to work. Hey. We can't hear them right now. They're chat. No, it didn't. So relay it to us. Tell us what that was said. Sorry about that.
Oh, what do you do with the DNA that you extract?
Ooh, that's right.
Yeah, so with the DNA that we extract, every organism, as we know, right, has DNA, and there is a certain part in this DNA that is specific for this organism. And so what we often do with the DNA, there are lots of different things that we can do, but what we often do, we look for this kind of like an identification piece of DNA, and we look for this piece, and then we can tell basically who is there and how many are there. And if you then have like this kind of incubation experiment where you have water where you put the oil in, and you have water where you don't have oil in there, then you can also see, okay, phytoplankton A is in the control water, but we cannot find it in the oil. And phytoplankton B might be in the control water, and there's way more in the water with the oil. And then you can see basically how we call it the phytoplankton community reacts to this change that we put into these bottles.
Great question, guys. I will note, too, for our students that are keen, we've been featuring this as a topic in several of our broadcasts over Oceans Week. eDNA is one of the coolest topics in the world. So if you want to do eDNA research in your life, this is like fairly new, and it's mind-blowing, the possibilities of this. So it's been a very exciting time to be a scientist. Miss Randall's class, you guys shared 2 questions in the chat, but more fun to have you live. So I'll come to our 7/8 virtual school. Hey.
Hi, thanks again, Jesse, and thanks, George. We really are excited about this. Stella wants to know, what's the biggest challenges our marine life face today? And I guess kind of how can we help them out? There you go. Biggest challenge. There's so many.
No pressure. Yeah.
Oh dear, that is what is the biggest challenge for marine life these days? I mean, it is like we still don't really know what will happen in the future, but we are very concerned about all the CO2 that gets into the ocean, right? Because that is causing the pH of the water to go down. So it's acidifying the ocean, and that might have like really like big impacts on, especially on the little phytoplankton. So that's a big concern. And then another big concern is the oceans are getting warmer and warmer. And as you saw, like in this profile that you showed, there are like different layers of water on top of each other. And for marine life, it's very important that these layers also get mixed around a little bit. And so there is a great concern that actually with the ocean getting warmer, that we have this, we call it is the stratification, that these layers become really stable, and then there is no mixing of nutrients anymore. And then there is concern that might very negatively impact ocean life.
Yeah. I'm really glad you mentioned.
And I mean, we also, I guess, have to say overfishing, right? I was now thinking about the little critters. But we definitely have to watch out what we take out of the ocean. And there were a lot of like plastic talks also, I think, right? All the plastic that ends up in the ocean, that's also a really big concern. So there are a lot of concerns about the ocean, that's for sure.
So it's a really interesting time to be a student growing up in this, because even in my generation, when I was growing up in school, we didn't really understand the full scope and scale of these problems. And it seems a little overwhelming to think plastics, bottom trawling, overfishing, habitat loss, climate change. I mean, it's daunting. The good news is that people under 20 pretty much universally know the facts, are keen to make a positive difference. And we had the chance to feature this Oceans Week and all year long, 500 broadcasts, I think 3/4 of which on conservation, that there are so many passionate people working to mitigate and change these issues for the better, which is great. So we are going to lose some things. It's not all peaches and cream for the next 50 years for sure, but the overwhelming tide is of positivity and hope, and I really think that that's a important thing to highlight in this. You're one of the first people to ever mention ocean acidification, and I completely agree, so thank you for that.
Yeah, I was more thinking of the little critters, but yeah, clearly there are more visible things, right, that are happening to the ocean, like overfishing, like microplastics, all that kind of stuff for sure.
We are going to do another round of questions because you're whipping through the Q&A, which is great. So St. Monica, I'm going to head back to you. If you want to share a question on behalf of your students or have anyone come up to the camera, you are good to go. Hey, grade 8s. Let's see. Take your time. No need to run. Hey.
Dagum, you had a question?
Eva, what was the longest trip you've been on?
What was the longest trip you've been on?
Yeah.
Okay, that was the 4 weeks. It was 4 weeks, and we started on the Cape Verde Islands. That's a little island group west of Africa. Very beautiful there, like lots of volcanoes and warm waters. And then we went to the equator. And we actually were just like circling around the equator a few times, because at the equator there are like upwelling events, we call them. So like cold water comes up from the bottom, and then there is a lot of nutrients in it, and there is a lot of stuff then that changes there, like phytoplankton starts growing, and yeah, that was the longest trip. And we actually we had a whole day at a station. It's called the 00 station, so 0 north, 0 west, and
Cool.
It was amazing. Yeah,
That's a really special, unique part of the world. We've almost never actually had people talk about expeditions there, so we love Gabon. We've done a lot of programs with land-based researchers there, so I'm really glad to hear all that. Miss Lada's class, you guys have a 2-part question, and I love it. So Kate wants to know, I'll highlight this, I'll bring you in. If you could not have your job, what would you do? And Maz wants to know, why did you choose your job? So why are you in this? And if you could change it, what would you be? Thanks, Miss Lada.
Wow, what would I change? What would I be? I think I would be a adventure photographer. I think that would be my other because I love taking photos. And it's great also to have this passion when you go on these research cruises. I have a lot of really nice photos from the cruises. And I stumbled like a few years back over the term adventure photographer. And I think that would have been something I would have loved to pursue.
Yeah. And then, of course, you got...
What was the second question?
No, it was sort of a rephrasing of the first one, how you got into it. So just being a boy, just super passionate about the oceans. And I'm glad you had those microbiology experiences when you were young, too. I hope our students get the chance to peer at things through a microscope, as it really does.
Yeah.
You know, when you use a microscope, when you look at a drop of pond water and see 1,000 organisms that live in that, and that's true of every drop of pond water everywhere in the world, you really can't help but be mesmerized by that. That is like black magic. It is so cool.
Yeah, I always say, you know, as fascinated as we are always about the space, right? The universe, like how big the universe is, and like these ginormous distances that are out there, like there is also no limit how small it gets. Right. So the world, when you look into the like how tiny things get, it's actually quite fascinating.
I highlighted on another one of our broadcasts, like every single one of you in your classes right now, as gross as you may find this, has little mites that live in your eyelashes, like all of you. Doesn't matter how clean you are, you have tons of little worms and mites living all over your face, everybody. And that is so cool. So whether it's something to look into the distant past, like telescopes and you know, grand scale, or the microscopic world, anything that extends the ability of our senses is a very cool way of exploring the world.
And the size range with phytoplankton is actually also ginormous. Like the smallest one compared to the biggest ones is basically like comparing the size of a fish to the size of the Empire State Building.
So sorry, I'm going to harp on this for 2 seconds, and then Miss Randall, I'm coming back to you to wrap us up. We're going to go long. We're being rebels together. But like, humans aren't used to this. We're actually a fairly big animal, and like people have a fairly close size range, even the biggest person compared to the smallest person. Look at birds, look at insects, look at ants. The smallest ant in the world, an entire colony of it could fit inside the head of the biggest ant in the world. And with phytoplankton, it's even greater. That is just such a cool thing to think about. Isidore, virtual school. I'm going to head back to you guys for one final question. Come on in and take us away.
Hi. So you answered our second question, but the second part of it, besides wanting to study in the field, you've told us how you're inspired for that. But we were curious, would getting a scuba diving certification be something that our students should be looking into, or what other interests can they pursue in their part-time outside of school to get qualified to do what you do for a living?
That's a good question. I mean, I actually don't have a scuba diving license or anything like that. It's really depending where your interest is. Like, but scuba diving, technical diving, science diving, these are also all things that would help you later if you are really like more like in the marine biologist field, right? I'm more like a cell biologist, so I do like most of my stuff is actually happening in the lab. But marine biologists are like more often out on field trips. Also, when you look at like seagrass and stuff like that. And for these kind of work, you definitely it's definitely beneficial to have, for example, like a scuba diving a license or something like that. Otherwise, outside. Yeah, what can you do outside the school to get into? I think just showing interest, just volunteering, just participating in, maybe there are like some citizen science projects, just going to the beach or the lake, just checking out what's going on there. Actually, like familiarizing yourself with what you maybe want to do later, and there are a lot of citizen science projects also. That you can be involved.
I love this question, and it is a tricky one to come up with on the fly, but it's been something we've been asked a lot this week. So again, scientists are always keen to help. You've never met a group of people that are more excited to share their passion and expertise and enthusiasm. So if you're keen on something, reach out, see how you can help. That goes such a long way to making a positive difference in your life. City Nature Challenge is a really great example of a citizen science project. The iNaturalist and CCAPs we use for a lot of our programs. Reading, being curious in general, go to your library. If you're fascinated with something, you can find so many resources, books, DVDs, movies, online stuff. It's just such a great tool. And for scuba dive interested people, and you too, Jorg, you should get your scuba dive license. It's pretty cool.
Yeah, I know.
Patty Bubble Maker, at 8 years old, you can start in the path to be a scuba diver, and by 10, you can have my certification, which is open water. It's like black magic to be able to breathe underwater. It is such a cool way of exploring 70% of the world. So a huge thank you for that question and for everything you share with us today, man. This is like the perfect capstone to what has been an epic National Research Council series. A huge thanks to them. A huge thanks to all our speakers for the Ocean Week. Jorg, thanks, man. This has been great.
Yeah, that was a lot of fun. That was great.
What we do to wrap up every broadcast, I'm going to bring in our teachers to say a big thank you and farewell. So St. Monica, I'll bring you on screen in a minute if you want to unmute your mic. Miss Lada, Miss Randall, thank you on behalf of your huge virtual classes today. You are all in the broadcast. Thank you so much. Have a wonderful day, everybody. Bye for now, guys.
Thanks so
See you all
much.
soon. Thank you. Bye-bye.